cells can be classified into two types depending on whether or not they are electrically excitable. So we have electrically excitable cells and electrically non-excitable cells. Majority of the cells in the body are not electrically excitable. However, sensory receptors are electrically excitable. They can pass on the information in the form of electrical impulses to neurons. So neurons are electrically excitable. Neurons pass on the information via electrical impulses, and they pass on this information to muscle cells. We have three types of muscle tissues in the body: cardiac muscle, skeletal muscle, and smooth muscle. Each of these muscle tissues have muscle cells, and these muscle cell fibers are all electrically excitable. There are a few other cells in the body that are electrically excitable. They're not very common. One of them is the endocrine cells that are found in the pancreas and the fertilized ovum or fertilized egg. They are electrically excitable. All other cells in the body outside of these categories are not electrically excitable. A stomach cell, a liver cell, skin cell, not electrically excitable. In order for a cell to be electrically excitable, it has to have a voltage that is very low. So a voltage from minus 50 millivolts to minus 90 millivolts at rest is very common for electrically excitable cells, whereas a non-excitable cell will have a voltage from minus 5 to minus 10 millivolts at rest, which is rather high and therefore not excitable. Before we study nerve impulse transmission, let's take a look at a simple reflex arc. Let's imagine that you're tasting something really good. The taste receptor picks up this information in the form of a chemical and passes it on to a sensory neuron. The sensory neuron then carries this information in the form of electrical impulses and passes it on to a motor neuron. The motor neuron carries motor information in the form of electrical impulses and passes it on to a salivary gland so that you may produce saliva. So in this example, the taste receptor, the sensory neuron, and the motor neuron are all electrically excitable. We mentioned earlier that for a cell to be electrically excitable, it must maintain a membrane voltage that is very low. So here is a neuron, there's the axon, and let's take a look at the membrane voltage of this neuron when it is at rest, which means that no information is passing through this neuron. Here is a section of the axon. The inside is negative in comparison to the outside. If we place a voltmeter on the inside of the axon, it will read at minus 70 millivolts, and we call this number the resting membrane potential because it is the voltage when the neuron is at rest. So how is the resting membrane potential maintained at minus 70 millivolts in the neuron? To understand this, we need to look at three rules. The first rule is that there is more sodium outside the cell and there is more potassium inside the cell. Rule number 2, there are sodium-potassium pumps all along the axon, pumping out 3 sodium and pumping in 2 potassium. The third rule is that there are leak channels, potassium leak channels, that allow potassium to move down the concentration gradient. Because there is more potassium on the inside, potassium ends up leaking out. So we have 3 sodiums to 2 potassiums going out. We have potassium leaking out. All of these are positively charged ions. So the net result is that more positively charged ions leave the axon, more positive charges leave the axon, and so the inside ends up being negative. So when you put a voltmeter and measure the voltage inside the axon when the neuron is at rest, the membrane potential ends up being minus 70 millivolts because the inside is far more negative than the outside. Because the inside is negative compared to the outside, we say that the membrane is polarized when the neuron is at rest. So our neuron is at rest. The axon, the dendrites, the soma, they're all at rest, and they all have a voltage of minus 70 millivolts. They're ready to pick up new information. This new information arrives at the dendrites and the soma, and once the information arrives, it changes the voltage in the dendrites and the soma, and we call this the local potential. It's called local because any changes in the cell membrane voltage is localized in the nerve cell body, mainly the dendrites and the soma. So let's see how that happens. What we see here in pink are chemicals called ligands. A ligand is a chemical that binds to something. When it binds to a channel, it opens the channel. In this case, the channel that we're going to look at is a sodium channel. It's a ligand-gated sodium channel. So let's take a closer look at that. Here is the membrane of the dendrite. The membrane of the dendrite has in red the sodium-potassium pump that we saw earlier and the potassium leak channel. Both of these are still functional. They still continue to do what they were doing. However, we have a third channel, and that is the ligand-gated sodium channel that is shown in yellow. This is the channel that needs to open to start the local potential. When the ligand, shown in pink, binds to the ligand-gated sodium channel, it opens the sodium channel and sodium rushes in. This is what triggers the local potential. What happens is that when sodium enters the cell, it brings along with it positive charges. When positive charges enter a cell, the cell becomes depolarized. The membrane becomes depolarized, which means the voltage goes up and approaches zero. That's what the word depolarized means. We call this the local potential. The membrane potential has now gone from minus 70 millivolts to maybe all the way up to 0 millivolts. The local potential can be really low or really high depending upon the strength of the stimulus. Again, the local potential is localized in the dendrites and soma. So what makes the local potential to trigger an action potential? In the local potential, we saw that the sodium enters the dendrites and soma via ligand-gated sodium channels. When they enter, the sodium diffuses across the nerve cell body until it reaches the axon hillock. When it does this, the voltage of the nerve cell body also changes, and the voltage spreads across the entire nerve cell body. These arrows represent the wave of depolarization that spreads across the nerve cell body, and it arrives at the axon hillock. The voltage has changed. It's no longer minus 70, it's probably 0 or minus 55 or minus 40, depending upon the strength of the stimulus. The minimum voltage needed to start an action potential is minus 55 millivolts. That's the minimum voltage that's needed. It can be higher, but it cannot be lower. We call this minimum voltage the threshold voltage. A threshold means an entrance or a doorway. In order to pass through the doorway and start the action potential, you need to have minus 55 millivolts, because when this voltage is reached, another set of channels called voltage gated sodium channels sense the change in voltage and automatically open. And the sodium that's outside rushes in, and that starts the action potential. So the action potential is triggered by the opening of voltage gated sodium channels. You want to imagine voltage gated sodium channels to be like motion sensors, those sliding doors that we see. When they sense motion or movement, they automatically open. And the location for triggering an action potential is the axon hillock. Let's take a look at the events that lead up to the formation of an action potential. We started with the local potential and the wave of depolarization that spreads across the nerve cell body. This depolarization arrives at the axon hillock. Remember, the axon hillock is the site for triggering the action potential. Now we need to check the voltage. Is the voltage high enough? If the voltage is at least minus 55 millivolts, which we call the threshold voltage, then the action potential will be triggered. This is because anything above minus 55 millivolts will be sensed by voltage gated sodium channels, which will open. When these channels open, sodium that has been accumulating outside the axon will rush in. When sodium rushes in, it brings along with it positive charges. And this causes the membrane voltage to go up. When a membrane voltage increases and approaches zero, we say that the membrane is depolarized. So the next step is that the membrane depolarizes. This depolarization is fairly rapid because many sodium channels open at once, lots of sodium comes in, and this causes the depolarization phase of the action potential. So the depolarization phase of the action potential is triggered by the opening of voltage-gated sodium channels. The range of voltage for the depolarization phase is from minus 55 millivolts to plus 20 millivolts. Let's do a quick review of the distribution of ions and the protein pumps and channels that are involved in the depolarization phase of the action potential. Remember, there's always more sodium outside the cell and there's always more potassium inside the cell. The sodium-potassium pump that is shown in red is always functional. The potassium leak channel that is shown in blue is also functional, allowing very small quantities of potassium to leave the cell. What we have here in yellow is the voltage-gated sodium channel, which triggers the depolarization phase of the action potential. During this phase, these channels are open and sodium rushes in, bringing positive charges into the cell and causing the depolarization. So our axon is now fully depolarized. Here is a section of the axon showing that it has plus signs on the inside and it is fully depolarized. The membrane voltage is now plus 20 millivolts. At this point, voltage-gated potassium channels open because they are sensitive to the change in voltage. Once the potassium channels open, remember that there is more potassium on the inside, so potassium rushes out. And when potassium rushes out, it takes positive charges out of the cell and the inside becomes negative again. So we say that the axon is now repolarized. We say repolarized because the voltage is going back to the resting membrane potential. So it returns to minus 70 millivolts. So let's see how that happens. So now let's take a closer look at how the ion distribution and proteins and channels work in the repolarization phase of the action potential. Remember that there is more sodium outside and more potassium inside. We're dealing with two different channels here. The sodium channels, which are the voltage-gated sodium channels, close, and the voltage-gated potassium channels open because they respond to the change in voltage. At 0 millivolts, voltage-gated potassium channels start opening, and by plus 20 millivolts, they are fully open, and potassium rushes out of the cell, taking positive charges out of the cell. This causes the voltage to go down. This is the falling phase, or the repolarization phase of the action potential, and the voltage goes back to minus 70 millivolts, or the resting membrane potential. Now let's take a look at the entire process using a graph. We'll start with the resting membrane potential, go on to the local potential, followed by the depolarization phase of the action potential, repolarization phase of the action potential, and back to the resting membrane potential. On this graph, on the x-axis, we have time in seconds. On the y-axis, we have membrane voltage in millivolts. We have minus 70, minus 55, 0, and plus 20 millivolts. At minus 70 millivolts, the neuron is at rest. We have the resting membrane potential. This stays the same until a new impulse arrives. When an impulse arrives, ligand-gated sodium channels open, sodium rushes in and causes a depolarization. We call this stage the local potential. The range of the local potential is from minus 70 millivolts to minus 55 millivolts. When the voltage reaches minus 55 millivolts, we call this the threshold voltage, it triggers the opening of voltage-gated sodium channels. Sodium rushes in and causes a depolarization, raising the voltage to plus 20 millivolts. At plus 20 millivolts, voltage-gated potassium channels open, potassium rushes out, causing the repolarization. The repolarization is so fast that it actually dips a little bit below minus 70 before the resting membrane potential is restored. So we have 5 stages in this whole process. The first stage is the resting membrane potential. When an impulse arrives, we have the local potential, which is stage 2. When the local potential reaches minus 55 millivolts, it triggers the depolarization phase of the action potential, which is stage 3. When plus 20 millivolts is reached, it triggers the repolarization phase of the action potential, which is stage 4. The repolarization goes all the way down and below the resting membrane potential, and we call it the hyperpolarization, which is stage 5. This stage is very temporary when the resting membrane potential is quickly restored. So let's take a look at what happens at each junction. We have A, which is where ligand-gated sodium channels open. B is where voltage-gated sodium channels open. C is where the voltage-gated potassium channels open. These are the main events of the nerve impulse transmission. So let's review these one more time. Stage 1, resting membrane potential. And the voltage is minus 70 millivolts. Stage 2 is local potential, going from minus 70 millivolts to minus 55 millivolts, triggered by the opening of ligand-gated sodium channels. Stage 3 is depolarization phase of the action potential. It ranges from minus 55 to plus 20 millivolts, triggered by the opening of voltage-gated sodium channels. Stage 4 is repolarization phase of the action potential. It ranges from plus 20 to minus 70, or maybe even lower. Stage 5 is the hyperpolarization. It is a very temporary state when the action potential is recovering back to the resting membrane potential. In order for action potentials to progress smoothly down the length of the axon, two requirements must be satisfied. The first requirement is that the action potentials must move down only one direction, that is towards the synaptic terminal, not backwards. The second requirement is that time must be allowed for the axon that is experiencing the action potential to recover from it and return to the resting membrane potential. So let's see how that happens. When the axon reaches the depolarization phase and completes the depolarization phase, it deactivates the sodium channels, the voltage-gated sodium channels, for a very short time. And in doing so, it sort of places a do not disturb sign to any new stimuli that are arriving at the axon. So that area shown in pink here with the do not disturb sign is called the absolute refractory period. The other area that is shown in orange says, do not disturb me, but you can if there is an emergency. Only disturb me if there is an emergency. So that's the period when the axon is recovering from the action potential. It's in the hyperpolarization phase, and it can only respond to a stimulus if the stimulus is very large. So we call this the relative refractory period. In this way, the axon allows the action potentials to move smoothly down the axonic membrane in one direction. Now let's take a closer look at the progression of the action potentials. The action potentials are progressing in one direction towards the synaptic terminal. Let's imagine we throw a flashlight and focus on the area that is being depolarized. So the inside of the axon is positive and the voltage is plus 20 millivolts. So this has completed the depolarization phase of the action potential. We say that the axon is depolarized. On the right, that part of the axon has not experienced an action potential yet. So that part of the axon is resting. And so we would expect the voltage to be minus 70 millivolts. And we say that the membrane is polarized. On the left, we have the area where the action potential has already passed. So that area should be recovering. And the voltage is going back to minus 70 millivolts. So this part of the axon is said to be repolarized. So far, we have looked at action potentials moving smoothly down the length of the axon. But this is not always the case. There are many neurons that are myelinated, and they send information much more quickly than unmyelinated neurons. So the myelin sheath here is shown in yellow, and this is a myelinated neuron. Between the myelin sheath are exposed areas of the axon, and these are called nodes of Ranvier. Each node has voltage-gated sodium channels and voltage-gated potassium channels that are exposed, which means the depolarization and repolarization can take place in these areas. So the action potential can skip large areas of the axon as it jumps from one node of Ranvier to the next, so that we see in red. As a result, it greatly increases the speed of conduction. And this is very useful for somatic motor and somatic sensory neurons that must carry impulses at a very rapid rate. We call this type of conduction saltatory conduction. Saltare in Latin means to jump, and this impulse jumps or leaps from one node of runway to the next. As a result, large myelinated neurons have the fastest speed of conduction.